start to
So we can change to the English.
Yes.
I'm recording the video, Behnam. If you just share your screen and then introduce yourself, then you can do.
Sure, of course.
Good, thanks.
Okay, let me share my screen. Okay, share. You got this presentation now?
Yeah, I could see.
Can you see that? Okay, very well. Okay, let me first introduce myself. My name is Behnam Razai. I am a master's student in the mechanical engineering faculty at the University of Tehran. I started my bachelor at 2014, and right after that, in 2018, I started my master. And it's about 2 years that I'm working on biomedical engineering field, and I hope that I can present to you today something that you can use, and it can be very helpful. Okay, let's talk about the field. Let's talk about the subject topic. Cell separation by deterministic lateral displacement. And what is that? First of all, I want to say, let's see what I'm going to say today. First of all, I'm gonna tell you an introduction and somehow read the scenario that why are we going to use deterministic lateral displacement. Then I'm going to tell you about its fundamentals, what kind of particles are separated through this phenomena. And you see in these two slides the titles that I've prepared to present today. If there is any question, I don't know, you can surely, Mr. Korban, please, if there's anyone have any kind of question, please you inform me. Thank you.
Sure, keep going. I will let you know.
Okay, thank you. Okay, so why is even cell separation important? What are the applications? Why do we even need to solve the separations? Maybe the most common reason is diagnosis. A very common example is cancer. You know, there is a stage in cancer. It is called metastasis. When metastasis happen for a cancer patient, the cells, there are some cells called circulating tumor cells, and they go through the blood to the distant sites of the body. And so the first application of cell separation is diagnosis. If we can test a patient's blood and we can find CTC in that circulating tumor cells, we can be sure that the metastasis has happened. And it is important to know in the right time that when metastasis happen. And the other applications of cell separation, which is now is very trending in the biomedical field, is cell manipulation and analysis. You know, there are a lot of examples. We want to somehow isolate some sort of cells. We want to manipulate them. For example, I think I told Mr. Orbani that I'm working on another project. We want to check and study the effect of some kind of enzymes on breast cancer. So we need that cell to be able to do cell culturing and this sort of things. So cell separation is important, first for diagnosis, and second, as I've written here, cell manipulation and analysis. Okay, there are some conventional methods. I'm not gonna talk about them. I've just written them here so that you can just be familiar with their names. Just a simple search in Google can give you some good details about these methods: centrifugation, chromatography, fluorescence activated cell sorting, and magnetic activated cell sorting. Okay, these are conventional methods that have been used for many years. I don't know how many years, but there are some problems with them. As I've write here, they need very skilled people in the laboratory. So what's the solution? Microfluidics, a new platform. It's been very popular in past two or three decades. Many people are using it. And why microfluidics are suitable, are proper for this job? Okay, let's talk about the reasons I've written here. Sample volume. You know, microfluidics, as it's clear from its name, the dimension of the devices of microfluidic devices are in the micrometer range, and so we need less sample volume, and this is very important. This way, if we need less volume, less sample volume, we will have less loss of sample, and of course less regions. You know, we have to add some regions to the blood or whatever our fluid is, like urine, saliva, whatsoever. The other thing is the ratio of surface volume force to surface force to volume force. And you know, I've not written the formula here because everyone comes from different field. Many people may not even be interested in physics or mathematics. Just it is important to mention that surface forces are here very much more important than volume forces, so we can use some surface forces like shear stress. There is less processing time, which is very good, and there are more precise. It has been approved, and it has been seen in many cases that its precision are better. So less, it is simpler, it is it is smaller, so it needs less cost for fabrication, and because they are small, they can be portable. So microfluidics is a good platform to do cell manipulation, cell separation, and this sort of things. Okay, there are two general methods for cell separation. One is active and one is passive. Because DLD, or deterministic lateral displacement, is passive, so I first say the active ones. Dielectrophoresis, as you can see here, excuse me. Dielectrophoresis, as you can see here, electrophoresis, acoustophoresis, immunomagnetic separation, flow cytometry, and optophoresis. As I told you before, these are not the topic of today's presentation, so I've just written them here so you can be familiar with these terms, and you can surely search and get a lot of detail and information about them. So what are their benefits? What are their cons and pros? Because you know, the definition of active methods is that they need external force for doing the manipulation, for being able to separate the cells, and this external power makes them bigger, makes them more expensive, and so makes them less portable. So versus these active methods, there are passive methods, and I've written them here: hydrodynamic filtration, pinch flow fractionation, or it is called PFF, inertial microfluidics, viscoelastic separation, and the last one, which is the subject of our topic, the subject of our presentation, deterministic lateral displacement. It is good to mention that it has another name, and you can see them in article, deterministic ratchets. Maybe there is a little difference between the deterministic ratchets and deterministic lateral displacement, but they're totally doing the separation based on one phenomenon, one fundamental. And this deterministic lateral displacement And all of these passive methods, as I've written in the right side of the page, they do the separation based on the channel geometry and intrinsic characteristics of the fluid. So they're less expensive and more portable because they're smaller. So after that, I'm gonna talk about the deterministic lateral displacement. Okay, why even we use deterministic lateral displacement? What is its benefits versus the other passive methods? You can see the first diagram, the left one, as I've brought it here. It can contain a good range. You see, the other methods have a more limited range than this. So you can totally manipulate particles, bioparticles, from the range of nanometer even to millimeters. This is maybe the most important reason that DLD is very widely used. And I'm sorry. It's me. Okay. The first time it was, I don't know, it was discovered in 2004 by Mr. Han. It was completely accidental. Its discovery was accidental. They were searching for ways to exploit DNA, and they were doing something else, and they understood that what they have designed can cause to cell separation. And what are the applications? The right picture in this slide, you can see there are two main application: particle separation and particle detection. Particle detection is much more new. I mean, the reports of particle detection is just from the articles from 2 or 3 years ago, 2016, and after that. But particle separation has been widely used by DLD from 2004. You see how does it do the separation. The most important way of separation is size. And the other things that are written here, it means when I say it does the separation by size, I mean some particles larger than a critical value go some way, and some particles smaller than some critical value go in another pass another way. And the separation have been done based on other characteristics like deformability, morphology, and electrical. So, what kind of particles have been separated by DLD? You see, all kind of bioparticles have been separated by DLD. The first one that I've already told you, circulating tumor cells or CTCs. And for every one of them, I brought just one example, as you can see, but there are, for many of them, there are more examples, more researches that have been done, more articles that have been published, and you can totally search them and see about the details of these jobs. Circulating tumor cells, white blood cells or WBCs, red blood cells, stem cells, parasites, spores, bacteria, exosomes, and DNA. You see the wide range of the dimension. For example, circulating tumor cells are very larger in comparison with the others. For example, it can be even 10 or 15 micrometers, but bacteria or DNA, you see the range can be in the nanometer range, their dimension. Okay, what is the fundamental of the DLD and how does it do the separation? Okay, as you can see in the middle picture, you see some circles. These circles are some obstacles against the fluid. There are some particles in the fluid, and this particle travel through this fluid, so fluid reaches these obstacles. And when it reaches the obstacle, as you can see in the middle image, that it's been somehow magnified, the colorful part. When the fluid passes the gap between two successive posts in a row, for example, look at the row I+1, and where there is a yellow circle, you can see that this fluid has been divided into 4 lanes. In the left image, you can see that 1, 2, 3, 4. So when and the fluid, when the particle is in the fluid, it is in one of these lanes. And if you can see, the first lane goes beneath the next obstacle, and the other go up on the upper side. So this way, the separation is done. I'll show you in the next slide exactly what I mean. In the right picture, you can see that the main parameters of its design. You know, there is a row, and between every 2 circles, there is a circle center to center distance called lambda, and the gap, which is here shown by the word G. And there is a lateral gap, and this is why there is lateral in the DLD. You see there is a delta lambda. We see a delta lambda shift from the previous row in every row. And as you can see in the middle image, you see that, for example, we have the row I, and the next row there is a shift delta lambda. Okay, the second row, I mean I plus 2, its shift in related to row number I is 2 lambda. And as it is, as we go forward, when we reach to the end, you see that again these circles are in exactly in front of each other. I mean I and the row number I and row number I plus n have the same position, and this means that the periodicity of this array is n. So let's call about the DLD modes and DLD parameters. So the relation of the shift delta lambda related to lambda is called epsilon, and it is called row shift fractionation, maybe the most important parameter in the DLD mode. For every DLD design, before everything, you gotta consider epsilon. And we have 2 different movement modes. Look at this image at the right side of the page. You see, if the particle is small enough, like the red particles, and if you want to know what is this, this is half of the post, the gray part, half of the post, and it is the fluid that is coming. If the particles are small enough, like the red ones, when they come, when they pass through this fluid, when they pass through this post, this obstacle, because they're small, and this contact force cannot overcome the drag force, so they go this way, they continue the flow lane path. But if they're big enough, they're larger than some critical value, like the green ones, you can see then when they reach the post and they have contact with the pillars, they change their path and they go from the lane 1, which is colored in blue, to lane 2, which is not colored. So we have 2 fundamental modes of movement in DLD. one zigzag mode. It's when for small particles when their diameter is less than a critical value, and another one is bump or displacement mode for when their parameters for when their diameters are larger than a critical value. Is there any question up to now, Mr. Borbani?
Actually, I couldn't see any question now.
Okay.
Just keep.
Am I clear?
Yeah, you can continue.
Okay, let's go. Before the design, as I've already told you, is the critical diameter because we want to do the separation. So the particles that are larger than a critical value go into the bump mode, and the one smaller than that value go into the zigzag mode. So we want to know about the critical diameter. And okay, there have been a lot of trials, a lot of simulation, maybe some formulas for the critical diameter. As I've already told you, I'm not going to enter the physical base of this phenomena. I've just brought this diagram, and maybe it is the most well-known diagram for the critical diameter. First of all, we want to normalize the particle diameter to the gap, because if the particle diameter is larger than the gap, it cannot even pass the gap. So you see this formula, DSC equal 1.4 G epsilon in power of 0.48, and this is the most well-known formula for the critical diameter. Mr. Davis has proposed this experimental formula. It was not done by simulation, it was done experimentally. And you can see in the right picture how many experiments he has done. For example, for the value of 0.1 for the epsilon shift fractionation, you see how many circles there are. And the circles that are shown in filled with black color, they have gone zigzag, and the ones that are not filled, are empty, have gone bump. So this way he could find the critical diameter and he could curve a fit. This is the most important formula for that. But something is important to mention, that this has been done for the rigid and solid particles. It has not been done for red blood cells, white blood cells, or this sort of things. It has been done for polystyrene beads that are very rigid particles, and it has been done for spherical particles. As you already know, many bioparticles, even red blood cell, is not spherical. So if we want to use the separation, this separation formula for the bioparticles, for the particles that are not spherical, we gotta use the effective diameter. How it is calculated, some simulations have been done, but it's mostly experimental, and there are some articles about that. Okay, there are two main design for DLD. First one is called parallelogram, and the other one is tilted square. In the left side, which is parallelogram, you can see the parallelogram in blue. The flow direction is normal to the gap. As you can see, the flow direction is in the vertical, and the center to center line is DL is horizontal. And there is another design which is called tilted square. And as you can see, they're not normal to each other. I mean, the flow direction to the related to the center to center line. The left one is more common, maybe because it is simpler to design, but the right one has its applications. Both of them are used, but I say in most of the articles I've read, the left one is used. Okay, there is a third. I'm sorry. There is a third motion mode, and it is called mixed motion. As I told you before, if a particle is smaller than a critical value, it should go zigzag, like the one that is written in, that is colored here in, I think, pink, and it is labeled A in the right picture. As you can see, it goes in a zigzag form, and the particles at the beginning of the path related to the particles at the end of the path have not laterally been displaced. There is no angle between them. The angle is zero. And as I've already told, there is a bump mode. And if there is a bump mode, we gotta see a full separation. In each lane, the particles should go to the next lane. But you can see in the B and in the C that these particles, even D, B, C, and B, these particles have not been completely displaced, nor they not zigzag. So there is a mixed motion, and it is completely calculated and seen by the angle, the displacement angle. You can see in the left image that, for example, Mr. Tanarak, his name is hard, he has done 3 experiments. He has done many experiments, and he has brought here 3 of them. For example, if dy here is the gap size, the G in the previous slide. For example, you can see the triangular diagram here, the triangular here, that its angle of displacement is 0, and in the circular one, which is not both the theta max or whether 0. So there is a third mode of displacement, and it is called mixed motion. Okay, maybe here is the most interesting part, deformable particles. You see, what I've already told up to now was about the solid particles, about the rigid particles. I told you about the polystyrene particles, but we really want to separate the bioparticles. Bioparticles are not necessarily spherical. And you can see here, I've brought research of Mr. Holm. He has done it on 2010. You see that this T. Silops, I really don't know the right pronunciation of this bacteria. We wanted to separate this. This is some kind of disease from the red blood cell. And when these particles are deformable, the flow rate of the device matters, because now if we increase the flow rate, there is more stress on the particles. It can change the radius. It has been seen that particles, when they want to pass through these gaps, they change the orientation. You see, they change that the smallest dimension becomes the effective diameter. So you can see this picture. Let's go to the next slide. You see, look at the right image on the upper side. You see when the red blood cell passes these pillars, its orientation changes, it's somehow turned around, and you see that its smallest dimension, which means 2.5 micrometers, is its effective diameter. But on 2010, Mr. Holm and his colleagues did some kind of novelty. They decreased the depth of the channel, so they made the red blood cell to pass through its its larger dimension. You see in the down image in the right side that when the depth of the device was decreased, it made the red blood cell to go by its large dimension, I mean about 6 to 7 micrometers, so it could cause that they can separate, as you can see here, these two. You see, the T cyclops dimension is about 2.5, and the small dimension of red blood cell is 2.5, so they could not do the separation. This idea of decreasing the depth, making the red blood cell go through its larger dimension, caused the separation to be done. And as you can see in the left image in this slide, that you can see the red blood cells on the down and the other on the upper side. Okay, there are some important things in DLD design. We should consider them. One of them is clogging. You know, clogging happens when the particles cannot pass through the gap. And okay, the first obvious reason is when the particle diameter is larger than the gap, so it cannot pass it. But sometimes these particles interact together. Sometimes they there's addition of them. They become populated, so they cannot pass the gap. And in this situation, we said that clogging has just happened. And you see particle-particle interaction, particle post-interaction. These are two things that can happen clogging. And what do I mean by particle post-interaction? You see, I told you in the first slides about the fundamental of DLD. And I said there is some kind of equation, some balance between the contact force and between the drag force. And when the concentration of the solution increases, I mean, there are a lot of cells in the fluid. So sometimes the other cells don't have some cells to interact with the post, and they cannot have a very this deterministic path. You know, when we say deterministic, we mean somehow we are sure about the path and the path of the particles. And when this happens, we see that it goes neither zigzag nor bump, and we cannot separate them very well. So it is very important to reduce the particle concentration. So we have to add some kind of reagents. One of them are called surfactants. I'm not very well aware about the surfactants, about the chemical formula, but whatever it is, they are called surfactants. They add it, so they dilute the solution. And this particle concentration, as I told you, because of these interactions, particle to particle, particle to post, can change the critical value of the diameter, and you should be very careful about the particle concentration. Okay, what I've already told was about the DLD, and in all the DLD designs that I've said up to now, the post shape was circular. So some people have tried to change the post shape to see whether there is a difference in, I don't know, throughput or in efficiency. For example, Mr. Lutherbeck changed the post to triangular, and when he did this, there were less clogging, there were less probability that particles cannot pass the gaps. And as you know, the triangular is not symmetrical. So in the forward pass versus the different pass, there is different critical diameter. You can use the very same device for two critical diameter. And so these are the advantages of triangular post. But it has its own disadvantages. It makes some kind of asymmetry. Asymmetry can cause some kind of irregularities, and it makes the prediction of the passes of the diameters harder. There is Mr. Beach tried the streamline, and in the streamline there is less stagnation points. By stagnation points, I mean the points where the fluid velocity is zero there. So the clogging probability decreases, and the eye shape has been tested by Mr. Zemin for non-spherical and deformable particles. There have been other post shapes that I have not brought them here, but you can see them in the main article that is on the poster. And the other thing is multiple separation. You see, if we want to do the multiple separation, you can see these designs A, B, and C, multiple array. I mean, in the first first image A. First of all, there is a separation. Very large particles get separated, go into different paths. Then the zigzag one reaches to the second array. So there we have changed the gap size, and the second the second time separation happened, and these sort of things. And there is Chirp and Cascade array. There are some differences. In the first one, the multiple array, this is very important that the maximum value of particle diameter is still less than the gap size of the last array. But if it's not possible, they use the Chirp array. And if you want to collect every iteration separation, and every kind of cells that they separate is important to them, they use Cascade array. And here you can see exactly what multi-cell separation. You see in the first stage of separation in the left side of the slide, at first platelets become separated from RBCs and WBCs. And in the second image, right side, they have entered a new array design. So then RBCs and YBCs, WBCs were separated. And there is something, maybe it has been talked about them less, but I've decided to bring it here. Because I myself have worked on it, and it's my thesis. It's in the process of writing, and it will be accepted. I hope it will be accepted soon. And you see, it's called about the asymmetry. In the main design of the DLD, we want to have a pressure difference just in the downstream way. Downstream pass. For example, if you look at the right image, the upper one, you see the flow direction. It is vertical. We want to have the pressure difference in just that direction. But when there is asymmetry for any kind of reason, there have been some reasons, they've mentioned it, we have different pressure in the lateral direction. And Mr. Verkener and Mr. Kulratarang again have studied this asymmetry. As you can see, I told you before, there were two designs, tilted square and parallelogram. And tilted square has no kind of asymmetry. You can see in the left image, the red dots, the red squares. And you see that there is no pressure difference for any epsilon value. But for the parallelogram design, which is more common, more usual, as I told you before, you see that changing the epsilon can cause to make some lateral pressure difference. And Mr. Kulratanarak came and did some change in the DLD design. You see here in the right side upper part, dx and dy. dy is the gap, but dx is not the gap, it's the distance between two rows. He increased this dx, and you can see he did it for one time dx equal dy, the other time 1.5, and one time 10. And as I told you before, when the fluid passes these obstacles, it is divided into some lanes. So if there is some kind of symmetry here, the flowing lanes that are being made should have the same widths, yes. And you see that when dx equals dy, it is not the same width. I mean, look at this image for dx equal dy. You see the first width of the lane is much more smaller than the last one. But if there is a symmetry, it should be equal. And you can see that for the ratio, for the value of 10, they are symmetry. So he concluded that for dx to dy equal and greater than 3, there is some kind of symmetry. I myself have come for my project, for my paper, have done some research on this ratio dx to dy, and could drive a new formula for a critical diameter when dx isn't equal to dy. Okay, up to now, let me look at the time. Okay, up to now, we have just talked about the separation based on the size, but the separation in DLD has been done based on the other things like the shape. For example, Streptococcus pneumonia, I say again, I cannot maybe pronounce them really okay, but they have been separated not just by their size, but based on their chain lengths. Mr. Bish did it about 3 years ago, and I've brought it here, the image. You see that the chain lengths, the ones that have longer chain lengths, have gone into the purple pass, and the other ones green and blue pass. And there has been some separation processes based on deformability. For example, when the blood is infected with malaria, it makes red blood cells stiffer, and when they get stiffer, when they contact the pillar, they are run away with more force, so they have higher migration angle. So he used this, and he did, for example, he could separate the RBCs that are affected by the malaria. And okay, I think these are the last things. Yeah. Particle detection. Up to now, we just talked about the separation, but in the recent years, 2018, 2019, just 2 or 3 years ago, particle detection has been done by this, and it has been done for nanobioparticles. For example, we know that if we send some polystyrene beads, some solid rigid beads in the range of micrometer dimension, we can simply predict their path. But when some nanobioparticles like proteins or exosomes, vesicles are added to these particles, they change somehow the particle characteristics, whether it is charge dominant, whether they changed its charge, whether they changed their size. For example, the coating of proteins changes electrical properties of the bit, so it changes the migration angle. So this way, there is some kind of calibration. They can understand how much is the concentration of the proteins in the solution. You see, if there is a lot of concentration, the migration angle will be bigger, and if there is less, the migration angle will be less. So they can predict the concentration of those particles, and this way they can detect it. And there is size-based, for example, when vesicles or exosomes, they are added to the particles, they change the particle diameter. So again, the particle migration angle changes, so they can understand the concentration of that. You can see Mr. Zemming article, and it has the details in that. And finally, finally, what are the challenges? So at the first, I told you that microfluidics have more throughput in comparison with conventional methods. What in passive methods, because there are a lot of obstacles in the DLD designs, there are a lot of resistance. There is more resistance in comparison with the others. So the throughput decreases. And by the throughput, I mean the amount of the blood or amount of the sample that can be processed at a specific time. So they have done some kind of trials for improving that. For example, they've changed the post-shaped sieve-based ones, and they have done some kind of parallelization. And for example, Mr. Da Silva parallelized 15 devices, so he could process 100 milliliter of blood in less than an hour. And another challenge is particle addition and clogging. I've already talked about that. I told you that some surfactants like PEG, Pluronic 2020, Twin 20 are added to the solution, so they decrease the addition ability of the particles, so they do not adhere to the posts. And I've told you they have changed the post shapes and gap sizes. And the other one is the complex and bulky experimental setup. For example, the DLD device may be in the whole chip, maybe in the range of some millimeters. You can completely hold them in your hand, but it needs at least 2 syringe pumps, and the syringe pumps are very big, and it makes their portability very hard, and it needs microscopes, and it is computer systems for data analysis. So it has made the DLD to be still yet more functional in the laboratory than in the marketing. And I brought an image that Mr. Salafi designed, something like a cell phone, like a mobile here, that can be portable and uses this microfluidic. I'm not gonna enter the detail because of the time. It's already about 6 p.m. And the fabrication process of microfluidics is completely very large, comprehensive course, and I'm not gonna talk about that. But just to point out, to say some points, it just uses it mostly uses standard lithography procedures, and most of the time they use PDMS, polydimethyl silicosane. I cannot remember the full name, or they use silicon or fused silica. And the last thing I want to say is that sometimes sometimes there have been some researchers that have added external forces. For example, Mr. Peach in 2009 used electrical field to change the critical diameter of the DLD. You can see in the right image that when the electrophoresis, dielectrophoresis force is applied on the particle, we can change the path and the way that particles move. And there have been some other external forces like mechanical strain or surface acoustic waves. I did not think that this lasts this much, but I tried to be useful. I tried to say as much as I know in this limited time. I hope you have enjoyed it. You could at least use a bit of information from my presentation. And I'm sorry for any kind of problem in my presentation.
Thank you so much, Behnam. That was a great presentation. And now we are open for question or suggestion. Also, we can change into Persian if you are like to do that, because I can't see any other people from the outside of the Iran. We can just keep going with the Persian because we can discuss more in Persian if you like. So,
okay.
yeah.